Okay, now in class we talked about a difference between spontaneous mutations and induced mutations, right? So spontaneous are just, spontaneous, right? So these are mutations that happen in our cells as a result of, say, imperfect cellular processes. Like sometimes purine bases just fall off, right? So they have that bond between the 5-carbon sugar and the purine base, which is less stable than the bond between the 5-carbon sugar and the pyrimidine base. And sometimes these fall off. So depurination, that's one spontaneous mutation that can happen. Loss of the base and then replication before it is repaired. It can lead to it is a type of spontaneous mutation, or something that's considered to be spontaneous. So replication slippage. So during DNA replication slippage, and this one, I like this one because it's kind of neat to think that, okay, replication isn't perfect, and when the polymerase is replicating a template, if we go over a repetitive area, so let's say we are okay, what are we replicating here? Let's replicate GTC. Okay, so here's our template, just repeats of GTC in a row. Okay, lots of these GTCs over and over again, a couple, maybe 50, maybe 100, who knows. So we're replicating this here. Our polymerase is traveling down this template, and it's putting in CAG one after another, and then the polymerase pauses. Oh, something happens. Something gets in the way. Something, who knows? There's a traffic jam, or it falls off. Who knows? Who knows? Whatever process caused it to something to go wrong. And maybe it fell off and needs to be loaded back on, and maybe these hydrogen bonds kind of separated for a second. So now we've got something that looks like this. Let me diagram this quickly for us. So I still have the template strand down there. So now we've got, okay, a loose little loop up here. So this is just a single strand, and when it reannealed, okay, now we had this loop come out. So okay, so I drew this a little bit longer than it was up here because I wanted to show you the loop, because imagine this is really long and I only diagrammed part of the repeat up here. So we have this loop here. Okay, well now polymerase gets back on and starts replicating. And okay, so now we have, when the polymerase is gone, long gone here, and this DNA is sitting here, this giant unannealed repeat area. Okay, so this needs to be fixed, right? So you have your repair machinery that's going to come in, and it's going to see all these CAGs. So one way they could fix it would be to cut this off somehow, right? Okay, that would work. But another way they could fix it is add bases down here. And I'm going to do that by drawing that in blue. And I'm going to make it a little longer. Okay, so we just added some more bases that complement those. And so we've introduced more DNA in here. So we had a repeat. Let's say we had a region with, let's say 50 GTC, let me CAG up here, CAG GTC repeats. Now we've added 1, 2, 3. We've added 3. So now, after replication and repair of the DNA, we have 53 CAG repeats, or CAG GTC repeats, we'll call them. Okay, and that can happen. Replication slippage seems to occur a lot in our repetitive DNA, even in humans. If we sequence these repetitive regions, we can see that these repeated sequences are shrinking and elongating throughout our genome. And I'm not sure what the mutation rate is exactly for these repeat regions in a single individual, but where humans tend to be very variable at this, no pun intended, at these repetitive areas when you compare sequences between individuals. Okay, so now let's look at the question. So a variety of neural and muscular disorders are associated with expansions of trinucleotide repeat sequences. The table below lists several disorders, the repeat motifs, their locations, the normal number of repeats, and the number of repeats in the full mutations. Okay, let's see if I can paste this in. Okay, there we go. So, a number of disorders: Fragile X, Huntington, myotonic dystrophy, I can't say that one, oculopharyngeal muscular dystrophy, and Friedreich ataxia. So we have trinucleotide repeats here, repeats of 3 nucleotides, and here the repeat is CCG, CAG, CTG, GCG, and AAG. Okay, and the repeat regions are located in different portions of genes. So here it's in a 5' untranslated region, so in the 5' ATR. Huntington, the repeat region is in an exon, so something that's actually, well, an exon can be a 5' untranslated region too, right? So I'm guessing by exon they mean the coding exon. 3' untranslated region, okay, so it's in the RNA, and it's after the termination site. So here for oculopharyngeal muscular dystrophy, we're in an exon, so I'm guessing this is a coding exon, coding for the protein. And then an intron here, so this is something that's going to be spliced out for Friedreich ataxia, but the repeats in the intron, and somehow that's having an effect. So we have a normal number of repeats. So people without these disorders, you look at their sequences, we have a normal number of repeats. For Fragile X, you can have as few as 6 CCG repeats, and that's so small that we can even imagine what that would look like, right? 1, 2, 3, 4, 5. It's not that small, I can't even fit it on my screen, but okay, this is what in one normal person, okay, not that many repeats here. I don't know if I'd rather have, I don't know how many repeats I have. I told you I did 23andMe sequencing. I don't think that technology measures lengths of repeats. Actually, I know it doesn't. But so some normal people can only have 6 repeats, and some can have 230 of these and still not have the disorder. So for people with Fragile X, they tend to have more than 230 of these CCG repeats in this 5' untranslated region of this gene associated with the disorder. So Huntington disease is, I'm getting a phone call. Hold on one second. Okay, sorry, I got a phone call. My wife was asking me where I put the electric pumpkin candles, because her and the kids have carved our pumpkins, and I'm at work right now doing these YouTube walkthroughs, and I don't remember seeing any electric pumpkin candles, so looks like I have some errands to run after this is the last video for today, so it looks like I'm going to pick up some pumpkin candles after this video. And so where were we? I was talking about Huntington disease, and so a normal person can have, not a normal person, a person without the disorder, and this is particularly cruel because you don't realize you have the disorder. Well, it doesn't actually have an effect until about 40 years old. but you can read about Huntington's if you'd like. Normally, it's a dominant trait, so it would run in your family. If you think, usually if there's no one in your family that has Huntington's, the likelihood of you having Huntington or carrying the Huntington mutation is very low. So, anyways, so a normal number of repeats is 6 to 35, and 36 to 120, you have the disorder. So, okay, so what we want to do now is let's take a look at the question. So most disorders attributable to trinucleotide repeats result from expansion of the repeats. Two mechanisms are often proposed to explain repeat expansion: unequal synapses and crossing over. So unequal synapses and crossing over. Okay, so we didn't talk too much about this because we didn't really in this course, this 200 level genetics course, we didn't talk too much about, actually at all, about the molecular basis of crossing over. So what scientists think is going on when you actually, exchange DNA between the chromosomes during recombination. We're going to save that for our class, BSc 350, the 300 level molecular biology course. So we're not going to worry about that mechanism, but we have talked about the second possible mechanism, errors in DNA replication, where single-stranded base paired loops are formed that conflict with linear replication. And so what we need to do is present a simple sketch of each mechanism. So we're not going to do the unequal synapses and crossing over. We don't need to worry about that. And we've already sketched the replication slippage mechanism right here. So we're already done with this problem. Or at least part A of this problem. So let's take a look at part B. Notice that some of the repeats occur in areas of the gene that are not translated. How can a mutation occur if the alteration is not reflected in an altered amino acid sequence? Okay, so where are they occurring where the sequence is not translated? 5' untranslated region here, 3' untranslated region here, and intron. So, well, what do we know about those 5' UTRs? Well, they're needed for some things, right? And we talked about this in lecture, and I hope it's in one of the lecture slides. And it won't be a question on the exam unless it's in the lecture slide, or on a lecture slide. But, so 5' UTRs have regulatory sequences, right? 5' untranslated regions, regulatory sequences where the ribosomes bind, right? For one, ribosome binding. So those ribosomes they just can't bind anywhere on the messenger RNA. They have to bind before the initiation codon so they can start translating the messenger RNA at the right spot and end up in the right reading frame and make the correct protein. So regulatory sequences in the UTR. We also have other sequences which we're about to see, I think, starting this week. We're going to take a look at some of the other regulatory sequences in these messenger RNAs that are important for the timing or translation rates. Let's say translation rates, and let's go stability of the messenger RNA. Stability of the messenger RNA. So messenger RNAs, once you make them, they don't last forever. There are proteins that degrade messenger RNAs, and they have certain half-life. So some messenger RNAs stay around longer than others, and a half-life is just the amount of time it takes for half of the messenger RNA of identical messenger RNAs to be destroyed. So they have different half-lives, and all that stuff is there are sequences in the 5' UTR that determine these sorts of things. Regulatory sequences, translation rates, stability of the messenger RNA, and thus half-life. So that's why you mess with that 5' UTR. Even though you're not changing the coding sequence, you are interfering with that protein. So and that can lead to a genetic disorder. Different disorder depending on what the gene is. Same thing for the 3' UTR. So the ribosome isn't binding there, but there are regulatory sequences there. The poly A tail is actually important too. Sequences that tell the ribosome when to stop translating, and things like that, and also sequences that are important for stability of the messenger RNA and also transporting the messenger RNA too. So also that goes for the 5' UTR. So there are sequences within the messenger RNA that are required for binding, for regulatory proteins to bind that messenger RNA and bring it to the right spot in the cell. So you can't always just put any type of sequence you want into these regions or have them expand and expect nothing to happen to expect to have no consequence on the translation of that messenger RNA. Even though they're not in the coding region, they can still cause problems with translation of the messenger RNA into the functional protein. Okay, for part C, in the two cases where the repeat expansions occur in exons, the extent of expansion is considerably less than when the expansion occurs outside exons. Present an explanation for this observation. So what that question is saying is, okay, so here it's occurring in an exon, and we already determined that the authors of the book must be talking about, they must be occurring in coding sequences. So even though 5' UTRs and 3' UTRs are found in exons, they're distinguishing between the fragile X mutations and say a Huntington mutation here by saying that these are coding sequences in the exons. And we can see that the number of repeats are smaller when they're in the exons than when they're found in the non-coding regions, like the intron here. Intron here, well, 37 to 1500, and the 3' UTR of myotonic dystrophy, and over 230 in fragile X. So why might that be the case? So here we're just speculating, and my speculation is, or my guess is, that so these are actual lengths of repeats. If you take people with these disorders and you sequence them, this is how many repeats you find. Now it could be that more than 120 repeats in Huntington is lethal. So if you have 250, if you have 500, and if you're born with that many, maybe you're never born. Maybe this leads to, say, a miscarriage or something like that. So it could be that more repeats in the Huntington repeat region, and the oculopharyngeal muscular dystrophy region lead to lethal conditions where the individuals just aren't born. So there's a limit to the amount of repeats that you can have with these genes and lead to a viable human. However, and there could be a limit because these are in coding regions. So these small number of repeats, relatively small compared to the others, they're longer than normal, but they're not so long that they completely disrupt the function of the protein, which must be important and required for viability. However, with the non-coding regions, it could be that longer sequences have less of an effect on those proteins. So they're still partly functional, even with the longer repeats, because those aren't involved in coding regions. And because they're not involved with coding, they're less important than the coding regions of Huntington and oculopharyngeal muscular dystrophy. Too hard to say. Okay, does that make sense? I hope so. So that's just one speculation, is that when you're not in a coding region, that the repeats have less of an effect on viability. But when you're in a repeat region for an important gene, when you're in a coding region, I should say, for an important gene, there's only so many repeats that these proteins can take before they become lethal to the organism, in this case humans. Okay, that's just one speculation. Maybe you guys can come up with some others. If you have some others, you can come to office hours. We can talk about them, and it's probably more than one explanation for these. So this is one of these spicy problems to get you to think more about what could be going on with using what you've learned in class and from doing the problems and applying it to human genetic disorders. Okay, so I think that's all of our questions for Chapter 15, and I'll see you in class on Tuesday.